All right, having been introduced to a number of big concepts in biology, we'll take an introductory tour of parts of the cell. Here we see two figures of the same cell. I'll start on the right. You can see this large area that's in blue. This is referred to as the cytosol. This is all the material that's inside the plasma membrane, but not inside an organelle. And lots of things occur in the cytosol, and we'll learn about those. Now going on to the left, these are the various parts that we will study and learn about in the class. The nucleus, the mitochondria, the Golgi apparatus, the endoplasmic reticulum, and so on. This is a slightly more detailed, more realistic picture of a cell, and we will use this to discuss the parts of the cell. I will occasionally refer to comparisons between a city and a cell, comparing the functions that a city needs to do and a cell needs to perform. This analogy was created by my predecessor in this course, Tim Gardner, and I find it to be a very useful analogy. Let's start by talking about the nucleus here, this double membrane enclosed area. This is where the DNA is kept. That is the information, the records, the archives of a city. You can also notice a portion of the nucleus that's stippled dark gray. That's a portion called the nucleolus. This is an area of specific activity. It's very high activity. It's involved with the manufacture of ribosomes, which once the ribosomes get out into the cytosol, that's where they'll make proteins. So this is an area of high activity. It's part of the nucleus, though. Here we see an electron micrograph where the DNA is stored. Here are the boundaries of the nucleus. Here are boundaries of a nucleolus. As far as I know, nucleoli are usually single-spaced rather than you see sort of two images here. The nucleolus might be extending beyond or underneath the plane that's that the electron micrograph was taken. That's a slice through a three-dimensional object. Not only is the information stored here in the DNA, this is where DNA is converted into an RNA molecule. This is a gene that gets turned on. This is utilizing part of the information for action. This RNA molecule will then get transported out into the cytosol, where it will be used to manufacture protein. The power plant of a cell is a mitochondrion. And there's many mitochondria per cell. I'll highlight on this one. You can see that there's compartmentalization within the mitochondrion. There's an outer membrane which encloses a white space, and then there's an inner mitochondrial membrane which is enclosing sort of a gold or a tan space. And on the next slide we'll see, or the next slide after this, just to highlight here's a fluorescence micrograph of a cell. We have one nucleus in a cell, but we have hundreds to thousands of mitochondria. Being the power plant, this is where ATP is produced. We have glucose breakdown products entering the mitochondria and being utilized to generate a high energy molecule, ATP, which then can be used to power many energetically costly syntheses or functions. Here's an electron micrograph of a mitochondrial. I'll start by pointing out in the figure here we have the double membrane structure with an outer mitochondrial membrane and an inner mitochondrial membrane enclosing the gold space. Here is a figure which is intended to correlate quite closely with the electron micrograph, and the main thing to notice from the original cartoon is that the outer mitochondrial membrane and the inner mitochondrial membrane are very close. The inner mitochondrial membrane will extend all the way almost across the cell, so that the inner membrane space is very small compared to the mitochondrial matrix, which is all the region inside here. This is the area that corresponds to the yellow area. The reason you have so much inner mitochondrial membrane is that most of the energy producing process occurs by proteins that are sitting in the inner mitochondrial membrane, and molecules that are in the inner membrane space will cross over into through the inner mitochondrial membrane. As the molecules get transported, we will capture some of that energy in the form of ATP, which then can diffuse out into the cell and power the various biochemical reactions that are necessary for cell growth. Now, protein synthesis can occur in two places. They can occur on ribosomes here in the cytosol, these dots, or they can occur on ribosomes on the endoplasmic reticulum. Proteins that are made on the endoplasmic reticulum are destined for the membranes or to be secreted to the external environment. Ribosomes in the cytosol are for proteins that are free in the cytosol or can get transported into other locations like the nucleus. As we already mentioned, the entire protein synthesis process starts where in the nucleus DNA gets converted to RNA. The RNA will diffuse out into the cytosol or be transported out into the cytosol, either to the rough ER or the ribosomes in the cytosol. Here we see an electron micrograph, and you can see the ribosomes dotting the endoplasmic reticulum, which is a membrane-based vesicle, and it's this dotted appearance from the ribosomes that refers to this portion of the endoplasmic reticulum as the rough endoplasmic reticulum. There's also a smooth endoplasmic reticulum, which we won't talk a lot about in the course. That is a site of lipid synthesis in the cell. The Golgi apparatus, or sometimes simply referred to as the Golgi, is a series of stacked membranes that are involved in protein sorting. We have proteins being synthesized on the rough ER. These will migrate to the Golgi, and then the Golgi will first they'll process the protein a little bit. They'll attach things like sugars and other post-translational modifications. They'll do a little quality control to make sure the protein is folded properly, and then it will package it in vesicles and sort the proteins. Maybe the proteins need to go to vesicles, maybe the proteins need to go to the plasma membrane, maybe the proteins need to be secreted and go to the external environment. This occurs in the Golgi. The plasma membrane is what separates outside from inside. It is the border of the city or the border of the cell. It's not only a barrier, it's also in charge of import and export of molecules that are outside that it wants to take advantage of. It'll export those into the cell, and it will also help secrete things. On this slide, we see the way that the plasma membrane does that. If there are molecules in high concentration in the outside world that the cell would like to use, they will collect probably on receptors sitting here on the protein membrane. They will be concentrated in a particular area. The membrane will invaginate, and then it will be pinched off, creating a vesicle, which will then fuse with other organelles inside the cell for processing. Quite frequently, the processing involves proteases chewing up proteins, nucleases chewing up nucleic acids into building blocks for the cell to use to make its own proteins and own nucleic acid. This process is called endocytosis, and we'll learn about this in the second part of the course. Conversely, the export process is called exocytosis, where we have molecules being concentrated in a portion of the endoplasmic reticulum, forming a vesicle with the contents inside. Like soap bubbles colliding, the vesicles can collide with the membrane. They will fuse, it will open up, the contents of the vesicle will be released into the environment, and then the curvature stress of the vesicle will be relieved, and it will tend to flatten out over time, creating some additional plasma membrane. Cells need a transport system. There are three parts that are highlighted here: microtubule and actin, which are involved in transport and structure, and then an intermediate, sort of the infrastructure, the intermediate filaments, which are less involved with transport. Let's talk first about the microtubules. These are the roads of the cell, and they extend radially from a centrosome, and if something produced near the center of the cell needs to get to the outside of the cell, and it doesn't want to rely on diffusion, a motor protein will attach to the cargo and essentially walk the cargo out to the periphery of the cell. Actin filaments, you've heard of actin and myosin in muscles, and actin is a molecule which can contract and produce motion. In this picture, you might imagine that the cell is moving in this direction. Actin is in many places, but can also be concentrated at the leading edge of cell movement. The intermediate filaments, in contrast to microtubules, are mostly structural, and they are mostly circumferential in the cell. These provide rigidity and strength to the cell's structure. These will all be talked about in the second part of the course. The cell needs a waste processing plant. The cell has two different types, peroxisomes and lysosomes. They have slightly different functions. Lysosomes are mostly an enzymatic waste processing plant. Proteases and nucleases will degrade proteins and nucleic acids in there into building blocks that the cell can use. The peroxisome is a chemical processing plant. Inside the peroxisome there's lots of aggressive reactive molecules, primarily oxygen radicals like hydrogen peroxide, which when you encounter a molecule in the environment, like a term that's often used as xenobiotic, this would be all the compounds we've learned to fear, plasticizer type molecules, drugs, other chemical compounds that the cell is not equipped to deal with. These will be degraded chemically and disposed of. We've highlighted that the nucleus is involved with information storage with the DNA. We've also talked about its role in protein synthesis, that's where DNA gets turned into RNA. This is also the region where the regulatory networks, the control center of the cell is. It's the place where information is integrated, and the proper response is determined by figuring out what genes to turn on in response to a new environmental clue. And the way environmental clues gets into the nucleus is by signaling pathways. This is the communication system of the cell. They typically will start with a protein on the plasma membrane binding an environmental molecule which gives it a clue. This information will be transported via a protein relay to the center of the cell, often by turning proteins on or off by phosphorylating them. That information is eventually transported into the nucleus, where a transcription factor might be phosphorylated, activating some genes in response to the new environmental stimuli. So that will conclude our whirlwind tour of the cell. I'll tell you that by the end of the course you'll know each of these parts intimately, and in the next set of lectures we'll talk about the molecular building blocks of the cell. Thanks.